Hi, this is Katie and Haley, and we are doing Lesson 8 group problem. So, Haley, why don't you take it away with number 1?
Okay. So for number 1, I'll just scroll down so you can see it. So for the normal chromosome, the LMN centromere is on the top of the synapse, the top of the crossover, and then the lowercase chromosomes on the bottom. And so when they come together, the bottom one has to make the loop because the letters have to match up so that they can exchange. I don't remember the exact reason, but they need to match up. And so product 1 is just the original chromosome. Oh my goodness, my computer's going crazy. And then product 2, you follow this line, start here, and you go over. And when you get to S, you cross over to T here, and then you just keep following the loop around. So you get to the end, producing a disymmetric. And then when you get the third product, I start over here because it just makes more sense in my brain. And you go from this side over, and you cross at the T to the S, and you keep following the loop around to get product number 3. And then product number 4 is just the original inverted chromosome. And that's how you get those products from that crossover.
All right. And so for number 2, for number 2, part A, we got non-disjunction produces 50% aneuploidy gametes, but meiosis 2 produces a monosonic phenotype, and this is because of how they split. Two of the 4 cells will have all of the chromosomes in them. Yes, 2 of the cells will have all the chromosomes in it, and in meiosis 2, one will have 4 types of chromosomes. One will have no chromosomes, and the other 2 will be evenly distributed. Okay, Turner syndrome is a monosomy because it removes one of the sex chromosomes. The familial Down syndrome is caused by Robertson translocation because it doesn't really have much of an effect. It's not very phenotypical. Okay, human aneuploidy can be seen by cells of N4, N8, or N16, so that would mean multiple copies of the same chromosome. Terminal deletion is common in people who exhibit Child-Chat syndrome, but intervision will cause type A hemophilia, which alters the the blood cells, which makes it hard to clot. A deletion will cause the structure to change in the chromosome, and this can be beneficial because it can get rid of a mutation that may otherwise be harmful. And it doesn't really alter the chromosome in an uneven manner. It makes it still line up correctly. Translocation is alternate segregation because they still can produce offspring. It just changes where the gene goes, but it doesn't mean that they become inviable. All right, and now we're on to going on to 3.
Okay. So for number 3.
Oh, I got 3.
You got 3. Take it away.
Sorry. No. Okay, so because Turner syndrome deletes one chromosome, you get 45 instead of 46. With Jacobs, it's 47, so you gain a chromosome. Oh, sorry. With Turner's, you lose 2. Is it 46? No, it's 46. That's normal. Sorry. So Turner syndrome, you lose 1. Jacobs, you gain 1. The Chateau syndrome, you keep it the same. The Familia, we changed that to 46, right?
Yeah, I just forgot to.
Okay. So with the last 3, it stays the same because it doesn't take away any of the genes. It just alters them. So they're still the same amount. They're just altered. All right. And then, so the only one that's not associated with the Marie tooth disease is duplication, because there's just not any duplication. The Kiefler, we use aneuploidy because, sorry, let me just pull up my notes real quick. Okay, because there is an alteration of the genes. It changes the number of genes that are present, and it's non-disjunction because it has a defect in the chromosome segregation. Okay, D. We weren't sure 100% on this, but we said aneuploidy. But now that I'm thinking about it, it might not be that.
I couldn't find anything because type A I thought was X chromosome inversion, so I didn't.
Yeah, that's what I thought too. It was inversion, but that's not one of them.
Yeah, so I don't know. I couldn't find any of those on the notes.
Maybe non-disjunction.
I don't know.
I don't know. Well, we'll stick with aneuploidy, but we are not sure why.
Sounds good. Okay, so number 4, that one's me. Okay. So it says 1 pair of homologous chromosomes in a mouse has the following sequence of alleles: D, E, F, G, H, centromere I, J, K, L, and another pair of homologous chromosomes has the following sequence of alleles. There it is. Suppose the reciprocal translocation occurs between 1 chromosome and each pair producing the following allele arrangement on 2 of the 4 chromosomes. So you get the DEFP-QRST on one, and then the MNOGHI-JKL on the other. Note, you still have 1 chromosome with the original 2 arrangements. And so this is how I set up the synapses of the chromosomes. So each of the chromosomes has to pair up with a like letter. So you have the 2 original ones here, and then I have the result of the reciprocal translocations. They're put here so that they can cross with the DEF right here on this chromosome pair, and N and O on this chromosome, and the rest on these ones and this one. And so for B, the composition of the gametes produced by alternate segregation. So by alternate, we crossed it across this way. So you get the 2 original chromosomes, which are viable, and then you get these 2, the 2 inverted chromosomes as well. And then for adjacent 1, we split it down the middle this way, and so 1 cell would have these chromosomes and 1 cell would have these chromosomes. And then for adjacent 2, we split it horizontally this way, so 1 cell had these chromosomes and the other cell had this chromosomes. Because when it separates, those are the 3 ways that it can separate when it's in anaphase. And then for number 5, cats having 38 total chromosomes would be found in a triploid cat. There'd be 57 because one of the gametes would have an extra chromosome in it, and so it would be the original number times 0.5 more, or added 1/2 more, so you get 38 times 1.5 is 57. And a cat, monosomic for chromosome 12, that's when you take away a chromosome because there's one, because it only produced a gamete with one chromosome in it. And then C, D, and E are kind of what Katelyn described earlier, where it doesn't change the chromosome count, it just changes the arrangement of the information on them. So the terminal deletion, chromosome 7 is still there, it just is a little bit different, so it still has 38 chromosomes. Same with the Robertson translocation between 17 and 18, they didn't go away, they're still there, so there's 38. And also with the same with the reciprocal translocation between 6 and 9, there will still be 38 chromosomes. They'll just be a little bit different. And those are the answers we got on the lesson 8 group problems. We still aren't sure about a few of them, but this is what we thought they were.
Okay. That's going to be it for tonight. Thank you.